Hi, everyone. I'm Laurie Hutcheson, Executive Director of the Lobular Breast Cancer Alliance, and I'm excited to be speaking today with Dr. Rinath Jeselsohn, who's going to tell us about the new Lobular Breast Cancer Research Program that launched at Dana-Farber Cancer Institute on Lobular Breast Cancer Awareness Day last October 15th. Dr. Jeselsohn is a breast medical oncologist and physician scientist who leads a breast cancer translational research lab affiliated with the Molecular and Cellular Oncology Division at Dana-Farber. Dr. Jeselsohn, welcome. And before I ask you specific questions about this new program, can you tell us a little bit about your lab at Dana-Farber, what you've been studying and finding, and the motivation besides our urging to launch this new program?
First, thanks for having me. It's really a pleasure to be here. So my lab is devoted to studying mechanisms of tumor progression and drug resistance in ER-positive breast cancer. We use preclinical models such as cell lines, but also patient-derived xenografts. And also we study clinical samples and to investigate what are drivers of treatment resistance to either endocrine therapy or targeted therapies, and try to identify new therapeutic targets to overcome resistance, and with the overall goal of trying to identify therapeutics that can improve patient outcomes. And then part of my lab, we also study lobular breast cancers as well, mostly from cell line models, but we're expanding now also to in vivo models, which is part of the research program of the lobular breast cancer here at Dana-Farber that we're very excited about.
Great. So can you describe the new ILC research program a little bit more?
So we've been really, over the past few years, have been really expanding our efforts in better understanding the biology of lobular breast cancer. And our group really studies a lot of the epigenetics of cancer along with genetics. And when we mean epigenetics, is anything that doesn't involve the gene itself or a genetic alteration. And as part of this, we were very interested in studying the estrogen receptor axis itself. And in lobular breast cancer, that's obviously a key axis. We know that the majority of lobular breast cancers are very strongly ER positive and ER dependent. And that led us to one of our first investigations of lobular breast cancers to look really at the estrogen receptor axis in lobular breast cancer. What we found first in clinical in cell line models, and now we have data also from clinical samples that really the estrogen receptor axis is different in lobular breast cancers compared to ductal cancers. In our preclinical studies, we showed that could affect response to specific endocrine therapies, but we don't know yet if that's really true in the clinic as well, in patient tumors as well. Which is something that we're hoping to continue to investigate and see really how is this different estrogen receptor axis really affecting response to endocrine therapies or other therapies that are related to endocrine therapies. So our initial studies really in preclinical models and mostly in cell lines really motivated us to continue to expand our investigation in lobular breast cancer and really open a program of ILC research program. And this is now expanded also to investigating, to building a cohort of lobular breast cancer patients, and specifically a cohort of patients with metastatic lobular breast cancer patients. So basically capturing the information we have from our patients here at Dana-Farber who've been treated here and have metastatic lobular breast cancer, and obtaining all the information on their treatments, their outcomes, and also we have genomics for all these patients. So we're currently, one of the studies that we're doing is really doing a comprehensive analysis of this cohort to understand how are the genetics in metastatic lobular breast cancer different, and how are the outcomes different, and specifically how are the outcomes in specific treatments in metastatic different in lobular versus ductal or non-specific, non-special type of breast cancer? How do they differ? And are there specific drugs that we see that patients with lobular breast cancer do better or worse in metastatic disease? So that's work that we're currently doing, and it's work led by Christina Fanucci, who is another clinician here at Dana-Farber, who's working with me on the lobular breast cancer program.
Oh, that's really great. So if I understand what you're saying, when you talk about a cohort, is this a prospective registry, or are you looking back
Yeah.
at or both?
Yeah, so right now it is prospective. So we basically it's real world data. So we basically put together a cohort of patients with lobular breast cancer, which is about 150 patients who have been treated here over the past about 10 years. We specifically collected, built this cohort using patients that we had genomic data for them from their first biopsy that was done in metastatic disease. And it was done within 3 months of treatment, first-line treatment in metastatic disease. So we really wanted to capture what are the genetics at the time that a patient is diagnosed with metastatic disease. So that is a retrospective cohort, but we're also hoping, as we expand our program, to also build a prospective cohort. And that's something that we're now working on building.
And so the retrospective is just metastatic lobular breast cancer
Yeah,
patients?
right now it's just metastatic. I mean, we have been doing, we've been doing genetic testing for the tumors using the OncoPanel platform that's here at Dana-Farber and Brigham and Women's Hospital. So that was the motivation to start with patients with metastatic disease, because we have genomics available for them. Because for patients with early stage disease, we're not doing genetic sequencing of the tumors at this time. But we also want to expand to early stage breast cancer patients as well.
I'm assuming that this cohort isn't all de novo metastatic, right?
No, it's not. It's all comers.
So then are you using the sort of, I know there's a lot of inconsistency from lab to lab about how to diagnose lobular breast cancer. And is that kind of across this cohort, at least, was the same methodology to determine the diagnosis used?
Yeah, so the advantage of the cohort is that it's all patients, their pathology has been reviewed by our pathologists here. So it has been reviewed by a small number of pathologists who work together and use very similar guidelines. So the diagnosis of lobular breast cancer is made based on the pathology review. But in addition, we have genomics. So we are looking at ecadherin mutations and loss of heterozygosity as well. And I can tell you that the vast majority have ecadherin mutations in this cohort.
Are you also going to be studying the subtypes of lobular?
So we unfortunately won't have enough patients in this cohort to really, do a rigorous analysis specific for the subtypes, but we do have that as part of our annotation. But I think as part of, as we move to do a prospective registry, we will be looking at patients with specific lobular subtypes and definitely annotating them, and hopefully we will be able to put together a big enough cohort that we can really better study their biology.
Oh, that sounds great. And I'm also curious whether, given this new lobular focus for research, Will there be any sort of corresponding an increase in clinical trial studies that are approved or embarked on by the rest of Dana-Farber that are more likely to have a be enriched for lobular or lobular focus than previously?
So as you know, we've conducted here one of the first trials that really was enriched for patients with lobular breast cancer and early stage disease. The PLAPS trial that we presented, the primary endpoints we presented at San Antonio this year at the lobular session. That is one of the first trials really of neoadjuvant endocrine therapy in looking at a subgroup of patients that were prospectively, identified as lobular breast cancer patients. And we are hoping that as we learn more about the biology of lobular breast cancer, we can open studies that are dedicated to lobular breast cancer patients. I think that now with the novel SERDs coming out, there is an opportunity now to really investigate how these SERDs, if they are different, specifically in lobular breast cancers that are highly driven by estrogen receptor. As I mentioned earlier, our data suggests that the estrogen receptor axis is different in lobular breast cancer, raising the question if these novel SERDs might be a little different in lobular breast cancers.
So I know that one of the barriers to participation in clinical trials for patients with lobular breast cancer and metastatic lobular breast cancer are the RECIST criteria that often excludes such patients. And I'm wondering whether in building the clinical trials now, there are some alternatives to, or in addition to RECIST criteria, so that patients with lobular breast cancer and metastatic lobular breast cancer will be less likely to be excluded from any clinical trials that Dana-Farber is conducting.
In many of the early stage clinical trials, we don't use response rates or objective response rate as our endpoint. Many times it is progression-free survival. And in many of these studies, we do allow non-measurable disease, which makes it a little bit easier to include patients with a bone-only disease. But clearly, I think this is nonetheless an issue. And then the other issue is the peritoneal disease and metastatic lobular breast cancer that is many times very difficult to assess, to really evaluate the full extent by the current imaging. As you know, we are starting to use FESPET more. We are opening now a study here that's sponsored by GE Health for FESPET, which is not just for lobular breast cancers, but I expect that a lot of the patients that we will be assessing as part of this study will be lobular breast cancer patients. But right now, I don't think there is a specific imaging that can really help us with this challenge. But hopefully there will be in the future other methods such as tumor fraction from ctDNA seems to be a nice surrogate to tumor burden, and potentially in the future this is something we will be able to use and could specifically also help for patients with lobular breast cancer.
Well, it's great to hear this is moving in that direction. I guess related, do you see any way in which the focus on lobular research at Dana-Farber will affect the Dana-Farber breast cancer treatment program at large? Do you think it'll be just generally increasing clinical trials or translational research?
I think so. Yeah, I think so. I mean, we are putting, a lot of it starts, I believe, a lot of it starts from the lab. I think that the science is what helps us, that is really where it starts from. And as we're dedicating a lot more efforts in the laboratory to studying lobular breast cancer, I think that is going to move forward. That's going to be an important part as we think about developing trials specific for lobular breast cancers. Also, like you mentioned, developing other methods to track progression will be important as we develop trials for lobular breast cancer. And also accurate diagnosis of lobular breast cancer is another part that's going to be very important at putting this all together. So I think these are all components that are really important, but many efforts are being made in all these areas. So eventually I think this will all, we will put this all together and be able to open trials specific to patients with lobular breast cancers and in collaboration with other centers as well.
That sounds great. So can you explain how in your research you include lobular breast cancer patients as advocates in your work formally?
So we have a very strong group of patient advocates at Dana-Farber, and for many of our proposals, they are a key part of this proposal, of many of our proposals, starting from grant submissions and as well as clinical trials. And their input is really valuable and very helpful, and many times this input really makes a big difference in what we're proposing and eventually what we're doing.
Are you able to use advocates kind of throughout the process of your study and then in writing up the results, et cetera?
Yeah, they are an integrated part of this process. Absolutely.
I have to confess, I'm a member of that group, and it is wonderful. But so glad to hear how much that it will be contributing to this new program as well.
Yeah, as we're building our prospective registry also, we are meeting with our advocates to have their input as well, which I think is key as well.
So I'm wondering if somebody wants to make a donation to support the lobular breast cancer research program at Dana-Farber specifically, are they able to reach out and restrict a gift for that purpose?
Absolutely, yes. Yeah, they want to donate to the lobular breast cancer program, and we would be more than happy to meet and show them the work we're doing in more detail, and where the gaps are and where funding would really help us.
I'm wondering if there's anything I haven't asked you about your new program, the structure, how you're proceeding, that you want to share that I haven't asked you about.
I did mention the PLABS trial, but I think that is also another key resource that we have right now. So this was a trial of close to 200 patients, and half of the patients do have lobular breast cancer. And tissue samples were collected from primary breast cancers at baseline, on treatment with endocrine therapy, and at the time of surgery after endocrine therapy alone or endocrine therapy with CDK4/6 inhibitors. This was a multi-center trial, which was a huge effort to get this trial done. And now the tissue samples that were collected as part of this trial is a huge resource for us that I think in the next 5 to 10 years, will continue to investigate to really specifically look at the biology unique to lobular breast cancer, is able to use clinical samples that have been treated pre and post treatment, and it's a really an invaluable resource.
Well, I think that wraps up the questions that I have for you. I really appreciate your taking the time to explain this program a bit more. We'll want to check in with you to hear how it's going and when and what is being published. And of course, as you know, and I think maybe I forgot to share that Dr. Jeselsohn is part of the LBCA Scientific Advisory Board. So we'll be keeping close tabs and look forward to hearing everything that you're studying and everything that you're finding. And we're so excited for this. So thank you.
Thank you for having me and for all the work that you do.